Alright, go Ezra. Before lunch. My name is Ezra. I work in the areas of Al-Kosviner labs, and I'm focusing on the interactions of Zika virus with Zika virus 3' UTR with host cellular proteins. So let's look a little bit about why we care about Zika in the first place. Zika is a mosquito-borne disease. Is this little guy? We're seeing more area where mosquito-borne diseases can spread to due to global climate change. We are getting an increasing range of the specific mosquito that carries Zika virus, which means that we're starting to see more instances of Zika infection in the United States, mostly in Florida and California. And why we care is that Zika has a tropism for tissues in the placenta that mediate the interaction between the mother's blood and the fetus's blood. And when Zika infects these structures, it can get from the mother to the fetus, where it will cause severe brain development damage. And it does this by infecting the neural progenitor cells. And you can see the normal brain over here with uninfected cells, and an infected brain where these neural progenitor cells are actually killed. And what happens from here on is that now these cells are dead and cannot differentiate into the normal parts of the brain. So you get a deformation called microcephaly, which is which is associated with severe developmental delay, seeing problems, hearing problems, and severe seizures and severe learning disabilities. So it's quite a socially and an expensive and socially damaging disease to get. So let's look a little bit at the actual Zika virus. It's actually quite a small and simple virus. Its genome is one strand of +sense RNA, which means it can be directly translated. Its coding region over here is flanked by two regions that are untranslated, or called UTRs. And these are kind of strange for a virus to have, because viruses like to fit as much genome or proteins as they can in as little space. So why is Zika kind of wasting some space on parts of its genome that, once again, aren't translated with the proteins? And let's talk a little bit about the biology central dogma. It's important to understanding how Zika replicates. Normally in biology we go from DNA, which is coded to transcribed to RNA, which is translated to a protein. But what we have in Zika is that we have a UTR that doesn't translate to a protein. Instead, the UTR itself folds and goes on to interact with host proteins. So let's take a look at why we want to look at UTR. It turns out that this UTR that we see in Zika over here, parts of the UTR are conserved all throughout the flatbee virus family, which suggests that there's some evolutionary importance to having this UTR in the first place, and that it performs some important unknown function. So we see that actually these two structures over here, they're found not only in Zika, but in dengue fever and Japanese encephalitis, which are more mosquito-borne flatbee virus. So previously, the Ugandan strain of Zika virus has been quite well characterized, and the first step was kind of to see how what the differences are between the Zika from Brazil, which is one of the main ones that we're seeing in America, versus the one in UTR. And here's actually the sequence alignment between the two. And as you can see, that other than maybe a few spots here, here, and here, that they're almost exactly the same. They're actually 96% the same. And if you go on, you can actually use that the Ugandan UTR, which has already been mapped out and split into its active structures, so 1, 2, 3, 4, and 5, is almost identical to the Brazilian UTR, which is the one that we're going to be talking about. And part of the project is a lot of molecular cloning. So you want to get out, you want to be able to manipulate the UTR and focus on different structures in the UTR. And an important part of that is knowing if these structures, if these structures expressed by themselves are stable. So what we have here is each of the parts of the UTR that you expect to find in the cell, with a little bit of modification at both its 3' and 5' ends. And these modifications are a result of the molecular cloning process. And we want to make sure that when we clone our UTR sequence, that it doesn't interfere with the folding of the UTR. Because the utility of the UTR is not just in its sequence, but also in its 3D structure and how it folds. And it turns out that they don't. So that's good. And here's just a little model of how I decided to split the active structures of the UTR, with one going from the 5' to the 3' end. So a large part of the methods, once again, is molecular cloning and how I must kind of pick the right parts of the UTR to study and then replicate them out. So we first start off with something called a PCR, which is DNA amplification, and turning it into a plasmid, or a vector, and then something called artificial transcription, which is how we actually get our active piece of Zika UTR RNA. So we start off with a template, DNA template of the UTR, and we attach primers to it. And these primers have a restriction site and a promoter sequence. Well, this one just has a restriction site. And what happens with these primers is the sequence gets inserted into the DNA template once that DNA template is replicated. So we get them over here and over here. And what this allows us to do is it allows us to treat it with restriction enzymes, which are enzymes that cut when they see a certain sequence. And we use our Zika 3' UTR template with sticky ends. And what we can do then is we can digest a plasmid or a circular piece of DNA with the same enzymes and actually insert our sequence into this plasmid. And we want to do this because this is more stable than just having a, RNA UTR to copy off of. So it's easier to store and manipulate DNA. And particularly in a plasmid, because we can actually transform a bacteria with this plasmid and create like a living genomic library of all the sections of the UTR we're going to study. And when we just want to, say, look at, say, section 1, we can pick the bacteria with the colony of section 1, isolate the plasmid, and then we can feature transcription. And here's my first cloning results. They're verified by a DNA amplification of the sequence that I inserted into the plasmid. So I actually took the live bacteria and put it into the PCR machine and amplified my inserted sequence. So that's what all these bars are. That's the actual inserted sequence. So the next steps are going to be in vitro transcription. And how this is done is that, remember before we put in the T7 promoter, what we use now is a T7 RNA polymerase. And that will start over here and create our piece of RNA, which is great. But now now the RNA is kind of in a solution with a bunch of other proteins. It's with polymerase, it's with free nucleotides, and whatever. And we don't want any of that. We want pure UTR. So now we must use a gel electrophoresis to purify. And what this does is it runs an electric current over our little samples that we load into the wells, and it separates out the RNA by electric current, and the RNA will move down to the electric current, or the electric gradient. And then after this, we can cut it out of the gel and then dissolve the gel and then get our piece of RNA. But that RNA is now non-folded because we just ran it through a gel. So much like pieces of unbaked dough, they're not really anything yet until we introduce them to the right conditions to fold, like you would fold and bake a pretzel. You would also have to now introduce your RNA to cellular-ish conditions, so neutral pH, optimal salt concentrations, and what have you. And that will encourage our now single piece of RNA to fold into our active UTR. And then the next part is identifying the actual interactions between the host proteins and this 3' UTR. And essentially it's like molecular fission. We have our now expressed Zika 3' UTR, and we want to identify what proteins will interact with it. So to do this, we first attach the 3' UTR to a little molecule of biotin. And then we attach this biotin to a streptavidin molecule attached to a metal B. And this is important because this is an extremely strong bond. So now this RNA isn't going anywhere. It's essentially anchored to this metal bead. So the next step is that we now would lyse some human cells, or any type of cell line that would be susceptible to a Zika virus infection, and run the lysate over this whole complex over here. And any proteins that would have any interactions with the 3' UTR will form a secondary complex. And then from there on, we can wash out the proteins that are attached to the secondary complex and then characterize them with Western blot and mass spec. Western blot will be good for identifying some of the interactions that we already know. We know that the UTR is involved in the disabling of the cellular interferon response, which is like the cell's way of fighting off a viral infection. So we would use Western blot for identifying if this whole process actually worked. And then after that, we would do something called a mass spec. And what that does, it gives us the sequence of the protein that is sequestered by the 3' UTR. And from here on, we can now characterize that protein. So basically, the protein complexes with the RNA will shed light on the interactions between Zika virus UTR and host cells. And lastly, I'd like to acknowledge some people. I'd like to acknowledge Diego and Carolina for helping me throughout all this project. It's been quite a ride. And I'd like to acknowledge Fritzy for helping me with all the issues that I've been too embarrassed to go and see Diego and Carolina about. I'd also like to thank the Gorman Scholars Program and CNSI for funding my research here and giving me this amazing opportunity in the first place.